Okay, so again, thank you for coming today. Welcome everybody. And obviously we're all here to see Peter, but before I hand over to Peter, I would just like to spend 2 slides giving a very short introduction to Biomedical to explain who we are and why we're here and why we're interested in single cell. So we're a small startup, we're based in Glasgow, and we are building an online platform, cloud-based platform for single cell RNA-seq data analysis. The important point to note is simply that this is what we're doing, this is what we're building. It's aimed at researchers, it is user-friendly, it's intuitive, it allows biologists to really explore their data and to generate publishable quality figures. I realise, of course, that a lot of this audience today are bioinformaticians and therefore have higher level of bioinformatics experience than the average biologist. So you might not think this is particularly interesting to you, but how we want to add value to your work is, for example, by providing a data management and data processing components that would simplify the data flow between a core facility and the end user, the academic user. So simply to say that we are Biomedical, this is who we are. If you're at all interested, please check out our website. And if you ever want to talk about your single cell analysis needs, please give me a shout and my email address is there. The other point to note is that we are recruiting right now. So we have positions available for a bioinformatician and for software engineers. So if you or your friends or colleagues or people in your network might be interested in these positions, again, just reach out to us. But you don't want to hear any more from me. The reason we're all here is to hear from Peter. I am not going to give one of those painful intros to his entire life story. I'm simply going to say that Peter, of course, needs no introduction. He is a leader in the field of single-cell RNA-seq analysis. And over to you, Peter. I believe you're happy to take questions at the end. Is that correct? You're on mute, Peter, just now.
All right, can you see my slides?
Great, yes.
Great.
So just
yeah,
to say,
so let's do questions at the end because I found that in online formats, this actually works best.
Perfect. Take it.
Yeah, so thank you for introduction, for opportunity to present some of our work. I was actually, I'm mostly going to focus on this kind of particular setting for single cell analysis, the case control analysis. But first, just a little bit of introduction. So I'm sure many of you are already quite familiar with single cell RNA-seq, but in general, the aim is to assess the state of complex tissues, right? So we can take a cell, so a little dot here, and measure its transcriptional state, right? So we basically measure the abundance of different transcripts in this particular cell. Of course, we can't get all the transcripts. We typically have a shallow sampling of the molecules that exist in that cell. Let's say about 10 to maybe 30% in some protocols. And that gives us an idea, a rough idea as to what this particular cell has been doing. Of course, single cells is hard to interpret, so we're going to measure a bunch of cells. And here, I've positioned cells close to each other if their transcriptional state, the measured set of molecules that we got from them, is somehow similar to each other, right? So we can start grouping them, and here grouping shown by colors. And these typically initially reveal distinct cell types that are present in the tissue, or perhaps different states. So by looking at these patterns, expression patterns associated with these different cells and clusters of cells, we can usually assign them some sort of a biological meaning, an annotation or cell type, for instance, and that gives us an impression as to what the composition of that tissue was. Now, if this is a continuous process, let's say a development or differentiation or something like this, we can measure more cells, and we're going to start to see these types of populations connecting the distinct clusters that we've seen earlier. This is sort of a way of sampling and view and looking at a dynamic process. And that too can be modeled computationally. So the most traditional way is to fit trajectories to these types of cell densities. So here we disregard these little empty cells on the top. We just focus on the bottom part. And we're fitting, each line shows a possible trajectory that's fit to the bottom side. And then so it connects, let's say, black cluster, pink cluster, and so forth. This is an approximation, right? So we're trying to approximate the path that the cells may take during differentiation. in the expression space, right? So that's supposed to reveal a sequence of expression changes that accompany this dynamic process. The interesting part here, of course, is that you could see some forks like this, right, where the cells that actually originate over here in the black cluster end up, let's say, on the left in the autonomic branch and mesenchymal branch, and so they have to pass this type of a decision fork, cell fate decision fork, and single cell data allows us to kind of look at this process, how this process is realized. You know, which genes are turned on, let's say, which transcription factors are turned on first during this decision process, and so forth. There's also ways of estimating, you know, the directionality of this dynamic process, because, you know, these types of trees or lines, you know, they simply tell you kind of the central axis along which, central path along which the cells may be going, but doesn't necessarily tell you in which direction they're going. And so some of our work allows to estimate this directionality. This particular picture that I'm actually showing you is from mouse neural crest differentiation. So this is a kind of a developmental study, an applied developmental study that was primarily focused on, let's say, cell fate decision making, right? Now, when we say cell fate decisions, in reality, we expect the majority of these cell fate decisions are not done by the cells themselves, but are done because of the influence of the external context. So I think one major direction that's developing now is analysis of this type of spatial context. So here we have an in situ measurement. So this is a slice through an embryo, mouse embryo. And again, we're looking at neural tube here in the center, and then here are neural crest populations that are moving off to the side, migrating. So this is an example of a snapshot where we see, you know, same transcriptional states that we've been analyzing before in this, you know, abstract expression space analysis, but now actually positioned physically, you know, and placed physically within the tissue, so we can start analyzing their context. And there are a number of methods that undoubtedly you've seen in the literature, particularly MERFISH, SlideSeq, and a few others such as SeqFish that are being developed for that purpose. So this is kind of just technical progress, and now I wanted to give you some examples of you know, where these studies are going, I think, with respect to clinically oriented investigations. So one approach is to simply, you know, look at, let's say, certain developmental paths or differentiation paths or normal tissue, and try to figure out features that actually altered in the disease, right? So in this case, again, we're looking at mouse neural crest, and this particular fork at the very bottom between the autonomic mesenchymal branch is actually reminiscent of what has been suggested of to happen in neuroblastoma, right? So in this case, we have a human disease, a pediatric cancer, and these are very heterogeneous tumors. And earlier studies have shown that they contain both mesenchymal autonomic compartment and suggested that the cells can actually shift between that. So to follow this up in collaboration with Nina Barniao from Karolinska Institute, we've actually sequenced a number of the neuroblastoma samples and tried to kind of integrate them into a joint analysis where we could see how this type of transition or connection between, let's say, mesenchymal and adrenergic compartments could happen, right? So this is trying to, again, follow up on the developmental study to draw a direct parallel between mouse development and intratumoral heterogeneity. So this, again, in this study design, you know, we only have tumors, right? And we're simply doing an integrative analysis of the tumors, and we're contrasting it with, let's say, normal development. In this case, even not even human development. So we mostly focused on profiling of the tumors themselves. Now, in general, I think it's fair to say that still the majority of the studies that are done today are these types of tissue composition analysis that the kind that I showed you in the previous slides. Most notably, let's say, human cell atlas efforts, HubMap, NIH HubMap initiative, as well as brain initiatives, they're all using these types of single cell techniques and spatial techniques to profile essentially steady state or healthy tissues. In the context of human cell atlas, they're also looking at development. And what will come out of this is this very rich atlas of cellular compositions, of cellular states, as well as some of the developmental differentiation trajectories of these tissues. Now, a separate design that I'll actually focus on for the rest of the talk has to do with the studies of disease, right? So in this case, we're looking, for instance, at cancer or neurodegenerative disorders, and the idea is that we're trying to find essentially targets, potential targets, and we do so through a comparative analysis, right? So pretty straightforward. The examples of this would be, for instance, Human Tumor Atlas Network, as well as the developing Lifetime Initiative in Europe. And when I say, again, it's a crude generalization, but when I say we're looking for targets, what is a target? So a target initially could be simply a cell type. Can we identify a very specific set of cells that is being perturbed, by the disease or influenced by the disease? It's much harder to infer causality, but we should be able to infer where the impact is. And because we're seeing the molecular description of these cells, the next thing we could do is to start analyzing, which pathways perhaps are most impacted, other transcription factors that are driving this, other potential external signaling events that may be related to this. So you could try to follow up and find, sort of not just cellular targets, but potentially molecular targets for follow-up studies. So the first example I'll give you, and I'll focus on it for the most part, is the comparative analysis we've done for bone metastasis of prostate origin. So bone is particularly, bone marrow, particularly common site of metastasis for a number of tumors, including breast, lung, and prostate cancer as well. So in castration-resistant prostate cancer, this is a nearly universal feature. It almost always goes to the bone. Unfortunately, there's a lack of effective therapies, and particularly the recent trials of immune checkpoint therapies did not appear to be effective, unfortunately, in the context of the bone marrow. One of the issues is that it's been kind of difficult to analyze, so we actually put together a team with surgeons, oncologists, and others to look at samples that were extracted as part of rare spinal decompression surgeries. So there, we can get these samples, both surgical samples as well as some additional samples I'll describe in a minute, take them through pathology and actually have high quality fresh samples of these metastatic tumors, again, to analyze their composition and to analyze what impact they have on the bone marrow. So the actual study design is that we take, in this case, a number of fractions, a number of sample types from prostate cancer patients, including this surgically removed tumor section. That's a tumor fraction, a liquid aspirate of bone marrow at the site of the metastasis itself, as well as sort of internal control, if you will, a liquid aspirate of bone marrow from a vertebrate distal to the site of the metastasis. So these are all matched essentially for each patient. Plus we have a set of independent samples from benign controls. So these are individuals undergoing hip replacement surgeries that have sustained inflammation in the bone marrow, but it's not cancerous in its nature. So all these samples, again, just by the experimental design, we take through single cell analysis, and that's what we'll primarily focus on to generate hypotheses that could be validated, in this case, in the mouse model. All right, so how do we go about this type of comparative analysis? In this case, we actually have 4 different cell bone types, but for simplicity, you could think of just the main contrast we want to see perhaps between, let's say, benign and tumor. So the first thing that needs to happen is that we need to identify the same cell types, let's say B cells, across the entire collection, right? So individuals vary, and I'll show you some quantification of this. And if you simply combine, let's say, experimental data, just count matrices or something like this from different individuals, the cells will be systematically distinct, right, by the expression profiles. And so instead of having, let's say, 1 B cell cluster, you're going to have like 4 or And so there are a number of methods for this type of alignment procedure these days. In essence, we're taking all these individual samples and combining them into a single, let's say, network. In this case, we use a graph-based approach that we've developed in the lab called CONUS. That establishes essentially a mapping between all the different data sets. And then when we produce an embedding, again, an expression space, an embedding that combines all these samples, then we can call clusters and essentially annotate them as cell types uniformly throughout the entire collection. So this is an integrated view. And again, this is a pretty well-developed area, so I'm not going into details. There's actually a nice review from Fabian Theis's group that came out, I think about a month or two ago, and I encourage you to look at it if you're interested in these integration methods. So this is an integrated view of all the fractions we measured for this study. And then the question is, so what's next, right? If we're trying to figure out, well, where was the focus of the impact of metastasis on the bone marrow, how do we go about this? Well, the first thing we can start looking at are compositional shifts, right? So it's pretty simple to understand that there will be certain cells that let's say appear to be more frequent, more abundant in one sample type than another. Now here I'm just visualizing the cells in a different color. So the cells from benign samples, benign controls shown in blue, and all the cancer fractions shown in gray. And what you can see easily is that, for instance, B cells are much more abundant in the benign controls. So for some reason in cancer fractions, we basically have a strong depletion of B cells. Now we could quantify this. So the easiest way to quantify this, of course, is just to take a fraction for each cell type. We calculate the fraction of the entire sample that it constitutes. And so here, different colors are different sample types, and each point is a sample. This is for, let's say, for the B cell result that I showed you. So B cells are quite abundant, notable in the benign controls. But if you look at any cancer fraction, they're barely present. Now, this is a very simple calculation. The statistics here are just a simple Wilkinson test, right? So we're not doing anything fancy. And the important thing that I wanted to illustrate is that even with a fairly limited number of samples, so we have about 10 samples, let's say, in each category, we're able to reach statistical significance across testing this 10, 15 different categories. So the statistical power is actually quite sufficient. The reason for this is that a lot of these compositional differences, they're very pronounced, like this B cell effect or the macrophage effect and so on. So even at these relatively modest collection sizes, you can still establish these trends with high statistical significance. There is one problem with this type of analysis, right? So if you imagine for a second that B cells used to constitute, let's say, 20% of the entire bone marrow, and in the context of a tumor, let's say for simplicity, all of them disappear. So if that was the only change, really, the compositional change that happened as a consequence of the disease, what you would see is obviously a depletion in B cells, but you would also see increased fraction across all the other cells, right? Just because we're basically calculating a fraction of the total. So this type of instability or dependence between these different estimates is well known, and there's an entire sort of little branch of mathematics that deals with this called compositional data analysis. So the main idea is that if you normalize your data in a different way, you can largely attenuate this type of dependence. So let me give you an example. Sorry, I'll go back to this slide again. But if I calculate the ratio, let's say, of T cells to everything else, right, when only B cells have changed, I'll see a difference, right? But if I'm calculating a ratio, let's say, of T cells to monocytes, or T cells to macrophages, or to any other cell type except for B cells, I'll probably see that they're not changing, right? So by calculating different fractions, we can actually find a pretty good explanation as to which cell type likely changed and which ones remain stable. So that's the general idea. So we basically transformed from this fraction space into what's called simplex view, which is a different normalization of these types of ratios. And then here, what I'm showing is just first two principal components of this simplex space. And the points are the samples. The cyan ones are the tumor, the pink ones are benign controls. And then the arrows here show the loadings as to which cell types actually configure this particular space. Now, what you can see is that the benign controls are on the upper right, and then tumors on the lower left. So they nicely separate. The loading of the space, so this is, for instance, immature B cells, right? What we expect. And here on this side, you have tumor cells and some of the other things that we expect to see in the tumor environment. So it nicely recapitulates the things we saw. Now, the question is, which of these cell types have actually distinguishing, let's say, tumor and benign in a robust way. So the way we assess this is we essentially model a separating surface here, and we're simply going to look for the loading coefficients on that separating surface within the simplex space. So that provides us kind of a nice intuitive view. So on the x-axis here, we have the separating coefficient for different cell types on that surface. And to assess significance here, we basically do a bootstrap sampling across both cells, right? So we take a limited number of cells from each cell type, but also across samples. A key thing, as we'll discuss, is variability between individuals in these analyses, and so it needs to be taken into account. So that allows us to estimate the uncertainty and hence the statistical significance of these separating coefficients. And in essence, what this picture tells you now in a much more robust way is that in the tumor fraction, we see increased macrophage abundance, increased tumor cell abundance, unsurprisingly, memory B cells, but not immature B cells and not mature B cells, for instance, right? So a pretty easy picture to follow. All right, so the flip side of more or less the same coin, on the same data, are transcriptional shifts. So we looked at compositional shifts, and now we're going to focus on transcriptional shift. And the idea here is that we can take a look at a single cell type, let's say B cells, and simply try to quantify the extent to which its expression state, on average, has been affected by the, let's say, disease. And what's shown here is just a prioritization of cells, right? So the cells on the right have this largest standardized distance, I'll explain it in a second, between tumors and controls, tumor versus benign. And the cells over here have the smallest distance. So the implication is that, let's say, progenitor PDC cells and CD8 T cells have been impacted, by the greatest amount, magnitude in terms of their expression state by the presence of the metastasis. This is meant to be a prioritization, so you could follow up on the most striking results. In this case, these two differ, but the rest are more or less at the same plateau. Now, what is standardized distance? Right? So there's an issue with measuring expression distances in general. So expression state, right, as you all know, is measured in this very large high-dimensional space, right? So we're measuring altogether, let's say, 10,000 genes across these cells. And so measuring distances in 10,000 dimensional space turns out to be rather difficult exercise. So if you use regular types of distance, like Euclidean distance, for instance, it simply loses sensitivity. So your ability to distinguish your nearest neighbor, let's say, and the cell that's least similar to a given cell, that ability becomes worse and worse as you increase the number of dimensions. So there are other distances that people use. Or they transform the space. So for instance, if you log transform the space and you focus, let's say, on a subset of genes, then that distance becomes slightly better, slightly more powerful statistically. Using, let's say, correlation distances on transformed abundance, also log transformed values, also appears to work better. Finally, we also use Jensen-Shannon divergence, which is kind of a distance between distributions, distributions of transcripts in this case between different cells. Now, none of these distance metrics are sort of biologically based, right? You could argue that one could be better for capturing some effects, and the other could be better for other effects. They also all have biases of a sort. So for instance, if you're looking at Jensen-Shannon divergence, it will be more sensitive to the changes in highly expressed genes, right? Because these carry more molecules and give you more opportunity to accumulate this difference. Whereas if you're doing if you're measuring distance, let's say using this correlation coefficient in the log transform space, right? The log transformation essentially gives much more weight to sort of lowly or medium expressed genes. So, if you have, let's say, transcription factors, which are commonly not highly expressed, then you would capture that better. Now, the different cell types that are shown here have different properties. The immune cells tend to be, fairly small, so that, let's say, they have lower coverage, lower number of molecules within them compared to something like tumor cells or endothelial cells. And so because of that, the bias of these distances will affect them to a different extent. The cells also differ in numbers. Some populations are bigger and you have more information on them, more molecules altogether, let's say, measured for monocytes compared to PDC cells. All of this creates biases that show up in these types of distance estimates. So what we use is a standardized distance, and what it is essentially a distance normalized by the interpatient distance within each category. So we take all the, let's say, pairs of tumor samples and benign samples, and we measure distances between them, right? And then we normalize it by the median distance within tumor collection and within benign collection. So we compare all the tumor samples between themselves and all benign samples between themselves for a given cell type. We find the median distance there, and then we normalize by this. That actually turns out to stabilize these results considerably and controls for most of the biases that I've described in terms of coverage and numbers of cells and so forth. Again, there's a cluster-free version of this, right, where you don't have to define clusters in advance. You simply look at, in this case, again, embedding space and quantify the magnitude of expression shift based on this. It is also possible, and I didn't describe it, for both density analysis and cluster-free shift analysis to calculate this not in embedding space, but in the graph space, right? So the graph space is the alignment between all the samples, and it is supposed to be a more faithful representation of how the samples related to each other. Whereas an embedding, like let's say UMAP, in this case large vis embedding, it will carry certain distortions. So for the most part, it's okay, but if you have very complex data sets, then you might want to keep in mind that the embedding is not sort of an absolute result. But at the very least, try several embeddings, to make sure the result is consistent. There are other ways of sort of prioritizing cells and getting an overview as to what the changes are. I won't go into all the different types of plots that we use, but here's just one example. So here we're trying to get an overview as to what is the functional direction of the transcriptional shifts observed across a variety of cell types. In this case, we're looking at genes that are downregulated in the context of a tumor. And what you have as columns of different cell types, what you have as rows are actually Go pathway clusters, right? So what we've done is we found all gene ontology sets that were significantly enriched in the set of top differentially downregulated genes for this cell type. And then if there were multiple Go categories that were essentially driven by the same genes, we clustered them and just show one name for the entire cluster. So as I'm sure most of you worked with gene ontologies, and the sort of hierarchical and complex nature of it is one of the issues, and we were simply using this clustering to provide a succinct overview of this. Okay, so what we see here is that there are certain Go categories that affect essentially multiple cell types, right? And then if you look at certain cell types, let's say these HSC progenitor population, stem cell population, it does have some unique features. For instance, strong downregulation mitotic of DNA replication, cell cycle, and so forth. You can do the same thing for upregulation. And you know, these types of, a couple of pictures give you a pretty good overview as to overall, you know, where different populations are moving functionally. Okay, so I'll follow up with just a few examples of actually what we find in this bone marrow study. So first, just looking at myeloid populations, we're just zooming into this corner of the embedding that I was showing you. And we can kind of subcluster it into smaller subpopulations to get higher resolution, and then try to characterize it. So again, all the annotations, as I'm sure most of you know, that are driven by sort of manual examination of expression patterns, certain genes, certain pathways. In this case, we use signatures, right? So we take literature-based or pathway-based signatures, let's say of monocytes, and then we simply run it across, you know, different subpopulations that we find. And so in this case, the mono1 and the TIM population, which I'll explain in a second, have clear monocyte-like signatures. Same thing for macrophage. So TAM population on the bottom seems to be a macrophage. Now, for the macrophages, again, we can score the expression of a M2 signature, which is a repressive macrophage state. And TAM, actually, this population on the bottom scores pretty highly. So that's why we call it tumor-associated macrophage. And for the TIM, the main thing we see is very high expression of inflammatory signature. And so we call it tumor inflammatory monocytes. Now, going back to sort of compositional changes, this is simply, again, this normalized density view across different sample types. And what you can appreciate is that compared to, let's say, benign controls, the tumor fraction shows very high abundance of both TIM, the macrophage population, as well as these inflammatory monocytes. All right, quickly shifting to T lymphocytes. So in this case, we took this entire T cell population and we reclustered it completely and generated different embedding just to get a higher resolution view. And in essence, the main thing we see is this is the density analysis. The main thing we see is that in the tumor context, we have a significant increase in these CTL populations, in particularly these are cytotoxic T cells, T lymphocytes, and particularly CTL2 population. And that CTL2 population, in essence, carries the usual markers of T cell exhaustion that have been profiled in other types of tumors. The overall relationship that we see is that especially in CTL2 population, is quite pronounced. So the cells that have and the patients that have higher exhaustion score have lower cytotoxicity in these cells. So this is kind of a classic phenotype of T cell exhaustion, where you have decreased cytotoxicity as well as upregulation of the specific exhaustion-specific markers. All right, so that's all fine. That's kind of just characterization of cell types. Now, if we're doing this comparative analysis, what else can we get out of this? Now, the thing to remember is that, especially in these clinical studies, right, that different individuals, first of all, they're impacted by the disease in a slightly different extent and different magnitude. And so, for instance, the abundance of this macrophage population or monocyte population will vary between individuals. In certain individuals, certain tumor samples, we see very high abundance, and others we see much smaller abundance. And we can use that variability to our advantage to try to answer some of the hypotheses or establish some sort of associations. In this case, we're primarily interested in trying to explain the exhaustion phenotype and decreased cytotoxicity in the T lymphocytes. So we can explicitly look for association between, let's say, the state of the CTL2 population in a given patient and the abundance of these macrophage population. And same thing here for abundance of these inflammatory monocytes versus cytotoxicity of the CTL2 population. And we see an association. Again, this is not earth-shattering p-values or anything like this, but then again, the number of samples we're really working with is not that great either. So the larger collections, as these studies become more accessible, collections with dozens of samples will give us much greater statistical power, and then we can examine more hypotheses and probably get more significant results. But the main message here is that there appears to be an association between the abundance of these myeloid populations and the state of this T cell population in the tumor. So it suggests that there's some sort of communication between them. And of course, the communication can be indirect, but it can also be mediated by specific signals, right? So there are a lot of cytokines that, for instance, are expressed by these myeloid populations, and maybe some of them are directly influencing either cytotoxic T cells or, let's say, Tregs that are in turn regulating cytotoxic T cells. So this then becomes a kind of a quest as to, can we identify the potential channel of communication that may be mediating this type of feedback? And so here, we're one idea that's been used in the literature is to try to basically match receptors and ligands. So we're going to look for, let's say, cognate pairs, where T cells have, let's say, a receptor, and then the myeloid populations express a ligand. And in general, if you take a look at any of these kind of signaling axes, using this kind of ligand receptor matching, you could identify, which populations could potentially talk through that channel. So in this case, CXCL3 CXCL3 signal can connect, let's say, myeloid 2 and NK populations. Well, that's fine, but if we take, a slightly more popular signal like IL-1B, there'll be more populations that can potentially talk through it. If you take interferon gamma, well, almost anything can talk through that, right? So altogether, if we put a table like this, and these are different T cell subtypes, different myeloid populations here, and here the number in the center shows the number of potential channels through which they could talk. And it's a very high numbers, right? I mean, we cannot possibly follow up on 400 channels, right? So we can make this more stringent. We can require that let's say the expression of a ligand and a receptor is seen in more than 10% of the cells over a certain threshold, and that can reduce the number of channels. However, we still need additional assumptions in order to narrow it down to something manageable, to, let's say, from like 10, 11 to a handful of channels that we could test. So other assumptions that we've made in this particular study is that we'll require significant upregulation of ligand in this TIM-TAM populations and myeloid populations in the tumor, right? So we require that, somehow in the tumor context, they need to be expressing more of this ligand. Receptor in principle, it seems unreasonable to require upregulation of a receptor. It could stay at the constant level and still transduce the signal. But we require for receptor to be not downregulated in T cells in the tumor context, at least. And that narrowed it down to this set of pairs, cognate pairs. And out of all of these, this CCL20, CCR6 actually seem to be potentially explaining both TIM and TAM signaling between TIM-TAM and the T cell population. So that's what we ended up following up. Those are the expression patterns of the ligand and the receptor. So receptor is primarily T helper Treg cells, but also present within the CTL2 population that is exhausted. And the ligand is expressed by both TIM and TAM populations at pretty high levels. And what follows basically is a mouse-based model validation. In this case, our collaborators on this have actually derived a model by optimizing a cell line through multiple rounds, a mouse cancer cell line, to select the cell line that specifically localizes to the bone, right, to model bone marrow metastasis as opposed to primary prostate cancer or metastasis in another tissue. And then there we could try to perturb that axis of communication, right? So we're talking about CCL20, CCR6. You could interrupt it. One way to interrupt it would be to generate a knockout, right? Knock out the receptor. So we did this, and then we see increased survival in the knockout mice. The other way to interrupt it is by blocking antibody. So again, we see increased survival. So now we tried to interrupt it from two ends, right? And then saw a similar phenotype. We can then go into these mice and actually figure out what happens with T cells when we interrupt this axis. And well, even here we could see that this population, which is actually contains all the exhausted T cells in the mouse, is decreased in the knockout. So that suggests that we're avoiding this T cell exhaustion phenotype. And then further validation of the protein levels actually for this PD-1 and LAG-3, so classical exhaustion markers. So we see them drop down to nearly naive levels. You know, these are mice that were not exposed to this tumor cell line at all. And then finally, a functional assay, so in vitro functional assay, where we take out these T cells, we stimulate them and see how fast they proliferate. So in this case, this knockout mice, you know, somewhere in between the naive and the wild type. All right. So the T cells, in essence, you know, when we interrupt this axis, the T cells show reduced exhaustion markers, right, and they're more functional, appear to be more functional. Anyhow, so that was it for the bone marrow study. Now just a quick summary on the sort of comparative analysis. And the whole process of trying to get from these type of sample collections to potential targets. So what we focused on is in terms of like tool development and so on, is disease versus healthy, right? Now it could be, you know, controls versus treatment and so forth, but basically a two-group design, which is the most common design, you know, in biology probably. In principle, if you have multiple groups, again, you could do these types of pairwise comparisons between individual groups. There are a couple of things that we think are common for initial steps. The cell type prioritization. So it can be done using these compositional shifts, so which cell types are basically changing in abundance. And the flip side of this are transcriptional magnitude or transcriptional shifts. And the reason why I say they're kind of complementary, because again, depending on how you annotate your clusters, right, the change might end up being recorded or detected as either compositional shift or transcriptional shift. So let's say if you had a disease that was impacting B cells in such a significant way that you essentially clustered them as a separate cluster, right? So then, you know, the difference would be in the compositional shift. That's where it would show up. However, if you did recognize that both of these populations were B cells, then you would see the difference in the transcriptional shift, right? So depending how you configure it, you know, the same effect can show up in either one. A key thing in all of these analyses is overcoming inter-individual variability. Here's just a quick example showing expression distances. So these are like cell type average expression distance within controls, within schizophrenia in this case. And actually, this is a trend that we see in almost all diseases, that the controls show lower variability than disease. And you know, this is probably an impact of the, you know, disease progression, of the treatment that these individuals receive, and so on. But in all these tests, the prioritization tests, the key aim really is to overcome this type of variability, to find, you know, what are the general changes, generalizable changes that can be attributed to presence of disease in this case. Yeah, so now some kind of, you know, more problematic issues. So the controlling for treatment and other patient covariates. So this, in these types of designs, is often very difficult. It's not an issue unique to single cell analysis, but for instance, in the prostate cancer, you know, all patients that come in with metastasis, right, to get the surgery, they all receive standard of treatment. So we cannot separate the effect of the treatment that they all receive, right, from the disease itself. We simply do not have access to other types of samples. And that's the case for many other diseases. So it's something just to keep in mind. Yeah, so this molecular prioritization, I talked a little bit about kind of pathway overview, pathway statistics. Obviously, from any of these differential expression analysis, you could get a list of genes and start following this up, as long as you pay attention to significance and stability of this result. And finally, I talked a little bit about cell-cell interactions. I give you an example of how we try to follow this ligand receptor matching, and discuss, you know, some of the limitations of this. I think, going forward, we'll be using, we'll be trying to develop, techniques that rely on other types of information, such as inter-individual variation, which I touched on a little bit, the actual modeling of the response signatures to different signals, as well as spatial analysis. The other thing I wanted to say is that actually all of these types of plots that I've been showing, so we're preparing a package that will implement all of this to just help other people do similar studies. It's called case control analysis. So, and unfortunately, it's not quite ready yet. We're hoping to release it this week, but it will probably take another week or so or two to actually package it up and release it. But yeah, I'll try. I'll make sure to announce it and maybe even forward it through the same email thread here. Okay, so that's it. I'm going to skip to acknowledgement. So this bone marrow study in particular has been a result of an effort of a pretty large group of collaborators, including oncologists. Oh no, go away. Including oncologists, hematologists, surgeons, and a lot of postdocs who've toiled over this, and students as well. So thank you very much. I'm happy to answer questions.
Fantastic. Thank you so much, Peter. That was brilliant. I suggest, considering that there are still 40 odd people on the call, I suggest that people type their questions in the chat box, because I can't see everybody at once. And whilst you're thinking and typing, maybe I can start with a question for Peter. So I really like this use of the variability between individuals to make observations and to generate hypotheses based on them. And obviously this relationship between the TAMs and the CTL exhaustion versus the TIMs and the cytotoxic phenotype. But can you overlay patient outcome on top of that to validate your hypotheses?
Yeah, it's a great question. Actually, we did try this. There's indeed seems to be some association there. Well, the outcome in this case, I mean, is response to therapy, right? So unfortunately, we don't really have effective therapies for these types of individuals, but some do respond to therapies better than others. The issue is that we just don't have enough samples to do this, right? So that's an important consideration for this type of interpatient variability based analysis. Basically, anytime you slice up your group, you stratify it, let's say, in responders, non-responders, and generate another hypothesis, you're essentially paying for it, with statistical power, because you have to control for the number of hypotheses you're testing, compensate for it, and so on. And yeah, in this study, even though there was a lot of work, but we're still talking about a couple dozen samples, right? Or at least actually it's about a dozen samples in a direct comparison like this, and that's just not enough to test a lot of hypotheses. That's why I think these types of approach is actually going to be increasingly more powerful as we're getting to sort of larger numbers. Even now, like the studies we're planning, they're sort of in 100 samples range, and then the same logic can be applied to a number of different hypotheses and maybe even some of the patient characteristics.
Yep. Yeah, that makes sense. Still no questions in on the chat. So again, I encourage everybody to think of a question and type it in the chat. I have another one standing by. So I was also interested that basically your outcome of the T cell exhaustion, you then used the CCR6 knockout blockade to show that the exhaustion was contributing to the cancer outcome. So why does checkpoint blockade not work so well in bone marrow metastasis?
Yeah, so that's a good question. We don't have an answer, right? And in fact, we don't know whether, let's say, even the axis that we're perturbing, whether it aligns or interacts with the standard PD-1, let's say, axis that is being targeted by these drugs. So that's something that we're hoping to uncover, or establish. But it's very difficult to say. It seems that there's I think, again, this is just speculation, but I think the process that leads to exhaustion of T cells is not a one-step process. There are probably multiple stages that are actually required for this. and what the perturbation that we're doing might be, the effect of it might be actually perturbing some of the earlier stages of exhaustion, for instance, that lead to exhaustion. It's quite likely that it is impacting, let's say, not the T cells directly, but either T helper or Treg cell activity. And in the end, it seems tempting to say that still the exhaustion that we see on the T cells is the mechanism that mediates this, survival difference that we see. But how we arrive to that state, that can be very different, I think.
Mm. Thank you. There are a couple of questions now on the chat. The first is from Thomas. So I think there might be a typo here, but let me just read out what it says. So why were all the clusters connected, monocytes to B cells, to tumors, I guess it's supposed to be? Is that an artifact or a true signal?
Yeah, I can jump in there. So this is hematopoiesis. That's happening in the bone marrow, right? So bone marrow is the site of hematopoiesis. So we expect all of this stuff to be connected, right? The HSC progenitor sits somewhere here. This is erythroid branch, monocyte branch, B cell branch. The T cells mature outside of the bone marrow. That's why they're disconnected. The tumor cells are indeed sitting close, and that was actually one of the questions that we got, why they're so close. They're not really connected. I mean, you can analyze this better. There's some, low quality cells that seem to be scattered here, and that's why it seems a little bit this way. But we also looked into the reason why they tend to be proximal. So it turns out that there's some similarity in terms of specific pathways between the tumor cells and the progenitor cells. Some of it has to do with metabolism, and so we try to control for this, for all sorts of ribosomal metabolic pathways and so on. Some of it has to do with cell cycle, and again we can try to remove this and control for this. But there are other features that essentially make these tumor cells slightly more similar in terms of their expression to the progenitor cells, even though they're of completely different origin. So that connection is not really a connection, but the rest of it is actually representing ongoing hematopoiesis in the bone marrow.
Great, thank you. A question from Ricardo: would pseudobulk approaches complement compositional and transcriptional shift analysis?
Say again, sorry.
Would pseudobulk approaches complement compositional and transcriptional shift analysis?
Yeah. So that's something I should include in my slides. The question is how do you run, let's say, differential expression? How do you if you wanted to find the expression difference between, let's say, control and disease, how do you do that? You have multiple individuals, and each individual has many cells, right? So if you simply combine them, then the individual that had more cells essentially would get a larger weight, right? And the biggest variability that we see realistically in these collections is between individuals and not between the cells of a given individual within the same cell type. So a very common approach for doing this, and the one we use as well, is this pseudobulk or mini bulk, where in essence you collapse all the cells from a given individual from a given cell type, as if it was a bulk RNA-seq measurement. So basically you use single cell data initially to do sort of an in silico sort, and then you purify, let's say, all the B cells from this individual, and you forget about the cellular barcodes. So all the molecules that you've seen from all these cells for a given gene, they'll add it up, and you get kind of a bulk profile. So that translates the expression test into essentially a familiar bulk setting where you have a bunch of profiles, kind of bulk profiles for control, a bunch of profiles for the disease, and then you can use standard techniques like DSeq2, LIMA, and so on to simply compare them. In fact, that's what we do internally. There are some caveats there. You need to worry about the number of cells that you feed in there. If they vary a lot between individuals. You also need to worry about stability of the results. So a simple way of assessing this is to do, let's say, a bootstrap subsampling or like leave one out types of subsampling on your patients, on the individual samples, omitting certain individuals, and then looking at stability of the let's say D results. The reason why I mention this is because a lot of times they are not particularly stable, and so if you're simply relying on the p-values, you might follow up on something that's not particularly promising. The alternative to this mini-bulk approach is a kind of a hierarchical model, let's say, where you take into account both inter-individual variability within each group as well as variability between cells within that group. These tend to be more time-consuming in terms of calculations, and at least in our test didn't really yield any benefits. They were not more stable, and they, the numbers of the genes were comparable. Yes. So, and the other thing I want to mention, just as kind of as a warning. So a lot of these integration approaches that I mentioned in the beginning, right? The ones that allow us to glue together all these multiple data sets, right, from multiple individuals. A lot of them end up giving you this kind of a common expression space, right? So they basically bring all the samples, they adjust the expression values to bring them into this kind of a common space. Not all approaches do this, but some do. And it's kind of tempting to take that adjusted matrix, right, and expression values to do downstream analysis. But that's actually not what you want to do, because like at the very least, this adjusted expression matrix will try to attenuate the effects that distinguish a disease and controls, as well as it will try to attenuate the differences between individuals. So if you run, let's say, a differential expression test on these values, you'll get inflated p-values, right, because the difference between individuals is attenuated. But also you'll have a reduction of the actual effect, right, because the difference between control and disease will be attenuated. So for almost all downstream analysis, except for visualization maybe, for almost everything else, you need to go back to the original values, right, non-corrected values. And that's when you'll see the actual variation, between individuals and between cells and so forth. Just kind of a word of caution.
Yeah, good to reiterate that point. Great. One final question, just on the topic of data integration. The final question is, some of these larger scale multi-center studies likely involve different centers using different technologies. So is it possible to integrate these different data sets that have been acquired, for example, on different technologies?
Yeah, so it's certainly possible. These integration methods, there are a lot of them nowadays. They've been getting a lot of attention. A lot of them are powerful enough to be used in different settings. So for instance, our integration method, we've shown that you could integrate different platforms, different species, even things like different modalities, like where you, for instance, you approximate expression from chromatin accessibility and integrate it with actual RNA-seq expression. It works, for some very tough cases, especially some of the interspecies comparisons, it will not work as well, but in many other cases, it will work just fine. The thing I want to point out is that all these methods, they're not parameterized. You never tell them actually what it is that you're trying to fix. They're all pretty much designed in a way where you just pile in the data and you say, integrate, and it will give you some sort of an integration. So if there are technical issues technical differences related to the platform, if they're minor, it will try to remove them. But the algorithms don't really know what they're fixing, right? So they can over smooth, that is, over align in a way where you lose distinction between certain subpopulations, or they may under align, right? Depending on the specific algorithm or setting. In that case, you basically have the platforms still clustering apart. So I would encourage you to try it, but just beware that most of these methods are kind of heuristic methods, right? So they're simply trying to find kind of the best match given the internal algorithm. But we do this all the time, like we're align 10x and SMART6.2, for instance. That's pretty common and usually works quite well.
Great. Okay, thank you very much. That seems to be all the questions, and I'm aware that we have run slightly over time. So thank you again, Peter, and thank you everybody for attending, and everybody enjoy your weekend.